Hi everyone, welcome to the Bioinformatics Coach channel. In this video, I'll show you how to map RNA sequence using the tool HISAT2. So for this tutorial, we are going to use paired end reads. And so what we are going to do in this exercise is to first install the tools that we will use. We will use Fastqc, MultiQC for quality control. We are going to use HISAT2 for mapping. And we are going to use some tools to do some processing of the output of HISAT2. We are also going to look at the example data we use. We will download it. I'll show you where to get them. That's fine. And then we are going to do quality control. We will now map the reads to a genome. So this genome here, we are referring to the reference sequence. And after that, we are going to look at the mapping statistics. So take note that when it comes to mapping, there are other things that can be done. For this tutorial, we are focusing on just these activities, and I'll leave some useful links you can use to also check things you can do post mapping. So for this tutorial, what you need to do is to make sure you have a Unix-like system, i.e. Linux or macOS. You should also have Fastqc, MultiQC, HISAT2, some tools installed. Alternatively, you can just have Conda installed, and then we will use that to install these tools. So whichever way, the instructions for installing all these tools can be found in the description box. So if you don't have Fastqc, MultiQC, HISAT2, some, don't worry, just make sure to install just Conda, and I'll show you how to use Conda to install all the needed tools for this exercise. So without further ado, let's move to the terminal and then start the exercise. Okay, so we are on the terminal now. So what we need to do is to first make sure we have our tools installed. We are going to use Conda to do this, and I repeat, instructions for installing Conda can be found in the description box. All right, so you can use Conda, or you can also use Mamba. All right, so either Conda or Mamba or Micro Mamba. If you have any of them, that is fine. All right. So in this case, I'm going to use Mamba because Mamba is faster. So I'll use that to install, but you can replace it with Conda. That's also fine. All right. So for now, let's create an environment and install the tools we'll use for this exercise. So I am going to install the tools. So I'll create an environment and install the tools. So I'll say Mamba create -n, and I'll say RNA seq. And then I'll say -c bioconda -c conda forge. I'll use these channels. Then I will specify the tools. So I need Fastqc. I also need MultiQC. I also need HISAT2. And I also need some tools. All right. So these are the tools I need to specify them. After that, I'm going to execute this command. And so what will happen is that Mamba, or if you're using Conda to whichever one, will, based on the information that you have specified, will get the appropriate packages for you, install and configure them for you. So let's wait for everything to get done for us. All right, so I have to type Y for yes. And after that, packages will be downloaded and installed for me. So I just have to wait for everything to get done for me. Okay, so the tools have been installed, so we can proceed. So I'll look at the instructions here. It says if I want to activate the environment, I have to use this command. If you use Conda, then instead of Mamba, you will see Conda. All right, so that's how you do it. So when you activate the environment, then you have access to the tools that you install. So I'll clear the screen and I'll activate. But before I activate, I'll just record this command again. I'll say that if you have Conda, then all I have to do is to change this and say Conda. And then still you have it created. If it's Micro Mamba, you just say Micro Mamba. So this will also get it installed for you. All right. But I use Mamba. So that's why it's this way. All right. Now I will just proceed. So I will activate the environment first. So to activate the environment, I will say Mamba activate RNA-seq. This is how I created it. So I need to use the same name. So this is how it's going to be. All right, it has been done. So I can even do a quick test. I can just say Fastqc -- version. Let's see. Just to test the tools. All right. So this is just to test. It's not really anything major. Okay. So I have everything I need. All right. So it's now time. So what we are going to do next is to now get the example data. I'll clear the screen first, and we are going to proceed. So for the example data, that's what we are going to use. So I am going to use data from this paper here. All right. So they have the data that they use for an RNA-seq analysis. All right. So this paper is a tutorial, by the way. So I'm only using the data here. All right. So you can read more about this paper if you have time. Okay. So the data itself has been compressed. And so to make it easy for you, I have prepared a script that we will use to get the data. All right. So the script can be found on this GitHub repo. All right. So I'll leave the link to this repo in the description box as well as the paper. All right. So when you get to this repo, I repeat, the link to this repo is in the description box. So when you get to this repo, what I want you to do is to get this script here, getData.sh. This script is what we need. All right. So what I want to do is to just open this getData.sh. So when you open this, what you see, what I want you to do is to go to this and then download it. You can also get the link. Okay, to download, you can just say right click and go to copy link. You get a link. We are going to download on the terminal, by the way. Or you can also click on this raw. I'm just showing you different approaches to get a script. Okay, so when you click on raw, you are going to get this link. All right, so I want you to copy this link. All right, so copy, that's the link that we will need. All right, to get the script. You can also just do a copy and paste, that's fine. Whichever one, it's okay with me. All right, so just copy this link and then go to the terminal. And then what we will do is that we are going to proceed. All right. So before we even proceed, we will need to also create a working directory that we will cd into it and work. All right. So I'll just say make dar, and I'll say RNA-seq. You can just do this way. You can even also say RNA-seq, whichever one is fine. It's just a name. All right. You can just say RNA-seq. That's fine. I can say RNA-seq. Let's use this. That's fine. So make that RNA-seq, and then I'll now cd into it. I'll say RNA-seq. All right. So now that I have cd into it, now we can now download the data. All right. So I'll just go over again. So to download the data, we just need to come to this and then copy this link here, this page. The link to this page is in the description box. So to copy it, and the data itself, this is the author. Okay, this is the paper that published data, but it's just for reference. But all we need is this link. So just copy it, and then when you come here, all right, you can now say wget, and then you paste the link to that script. Okay, the script that has the information. Okay, that has the commands to get the data and then process it. So this is a script. All right. So wget, and then you specify the link to that file. You can also use scale. That's fine. So I'll just execute the script to make sure I get the file. So now if I do an ls, I'm going to see the file, which is here. It says getData.sh. This is the file. So it is this file that I'm now going to use to get the data that I need for this exercise. All right. So what we need to do is to now execute the script. So before we execute the script, let's add execution right. So I'll say chmod +x, and I'll say get data.sh. So this will make it executable for us. Okay, so now that we have this, what we will do is to now download the data. All right, so again, I'll just take you through the commands. I'll go back to the GitHub page. I'll just go to this section. So basically, there's a command that gets the data and then it decompresses it for us, and then it now does some processing, okay, to make sure the data is arranged nicely for us on our computer. So that's what it's basically doing. Okay, so I want you to just focus on getting the data with minimal effort so that we can proceed with the main activity. All right, so let's execute the script. So I'll say /, now I'll say get data.sh. All right, this approach. So we will use this to execute the script and then get the example data. All right, so this is what we are going to do. I'll just redo it here. I'll just clear the screen first, do an ls, and I'll redo it again. All right, so I'll say /get data. This is it. All right, so this will get the needed data for us. So let's execute the script. Perfect. So once we do it this way, it's going to download and then do the necessary processing for us. So this will take some time, depending on your internet speed, but it shouldn't be that long. All right, it shouldn't be that long. So just be patient and wait for it to get done. So the total size is like 2 gig, so it shouldn't take much time. All right, so it's still working. Okay, now it's done. Perfect. So once it's done, you will see all these tests, okay, coming up, and then you see this, and then you see the report here. Okay, so now that we have this, let's clear the screen and then let's do an ls. So when we do an ls, you are going to find the data which is here, and then the ones we will need for this tutorial can be found here. So we have a directory called Fastqc. We have a script called map.sh. We also have this directory called ref. Okay, so if you ls into Fastqc, you are going to find the files here. So we have multiple files here. So these are paired and reads. So we have some samples here. Okay, and then we also have the map.sh, and we also have ref. Let's ls into ref, and then you are going to see it here. Okay, so this is what we have. All right, so we are going to see something like this. So let's clear the screen and then continue. All right, so we will look at the input files again. So when we do ls into Fastqc, we see our files. All right, so these are the files. Now let me see that the data here, there's a subset. Okay, so that's why you see CHRX1, 2. So that means that they only presented the chromosome X. All right, so there's a subset. But if you want the full data sets, these are the accession numbers. So you can search in the SRA database, or you can also search in the ENA database. You'll find the entire data set, the full data sets, in case you want to use that one. Okay, so this has been subsetted probably because it will be faster to run the analysis, especially for those who have limited computing resource. Okay, now if you have your Fastqc files, one of the things that you want to do, or you want to start with, is to do quality control. All right, so quality control allows you to assess the quality of your data. It can also help you to identify issues earlier so that you can resolve them, and so that it allows you to run your analysis smoothly in the later stages, okay, of your pipeline. So we are going to do the same thing. We are going to start with the QC. to assess the quality and see if everything is okay with what we have so far. So that's what we are going to proceed with. So to do the QC, what we are going to do is to run Fastqc first, and then we will run MultiQC. All right, so these are the two things we are going to do. So Fastqc to get us the initial report, and we will aggregate everything using MultiQC. I've covered some tutorials on Fastqc and MultiQC, but we are going to do something now. So this one is for RNA-seq, so take notes. Okay, so to run Fastqc on your example data, you just say Fastqc, and then you specify where your files are. In this case, it's in the Fastqc directory. So I will say Fastqc /star. I can also add .fastqc.dz to be specific. So that means all files ending with .fastqc.dz will be analyzed. So let's execute this command. So once we do it, Fastqc will start its analysis and it's going to run for you. Okay, so this will take some time because it's going to take them file by file. There are ways to kind of reduce the computational time, and it's something that for those who are interested in can let me know later on. All right, I'm also working on a Nextflow script to automate this process, so in due time I will share that on this channel, so make sure to come back as well. Okay, so Fastqc has done its work. So let's clear the screen. Let's ls into the Fastqc directory again. And then we are going to see these files. We have HTML and we have zip. All right, so these are the Fastqc reports. Now, if you have several files like this, it's very cumbersome trying to read them one by one. So we are going to aggregate all these reports into a single one, and we can read that one. So to aggregate the Fastqc reports using MultiQC, we say MultiQC, and then we specify the directory which has the file. So i.e. Fastqc / and we say star Fastqc star. That means we want just the Fastqc reports. So now let's execute this command. So once we do it, MultiQC will aggregate the reports, it's fine, and then it will let you know how it saved it. So it says found 24 reports. We have data, MultiQC data, reports, MultiQC reports. And so this will be the final outputs, and we can examine them. All right. So what I want us to do is to look at the files and then have a discussion. So this is the MultiQC reports. All right. If you want the individual reports, they can be found here. This is what we have. Okay. Now let's start with the individual reports. We'll just look at one of the files to explain a couple of things, and then we can look at the MultiQC output. So let's pick this one, for example. This is read one for the sample. It says ER 188044 CHRX1. Okay, so let's just use that to explain some things. Okay, so we see the report here. All right, so if you take a look at the report with Fastqc, when you have your analysis done, all of them will be listed on the left side. We see them here, and then we have those that have the tick, the checked symbol, and they also have a color green, meaning everything is okay. There are those with exclamation mark. That means something slightly unusual has been observed. And those ones will have this color, I think it's orange or so. And we also have those that have this X symbol, meaning there's danger. Okay, it's red. That means something unusual has happened. Okay, and so without even going through these reports, these color schemes or symbols should give you an overview, and then you can now start looking at those that are of interest to you. So from here, a lot of the things are okay, they are looking good. We also have basic statistics here. Here I will just look at the sequence length. It says 76. Illumina reads are short, so this makes sense. All right. Now, I will focus on just those two here, these two which are indicated here with the X, those that have the color red. All right, so far the other stuffs I will ignore them because I think they are okay. So I will skip them and we'll just talk about these two. All right, and then for those who want a detailed explanation to Fastqc reports, I've made some tutorials and I also give you some materials you can use to read more about them. Okay, so just take note of that. So let's look at the per base sequence content, this one. So when you click it, you will see something like this. So from the Fastqc manual, the per base sequence content is a plot that shows the proportion of the 4 DNA bases, i.e. A, T, C gene. All right, so a good data will have an even distribution. All right, okay, so this will be shown as parallel lines. So we have 4 lines, each of these represents one base. All right, so we should have parallel lines. We see the parallel lines here, and then we see those that are wobbly on the side. So when we have an observation like this, where we have these lines being wobbly, for example, here, it indicates some bias. That means there is a bias, okay, in the data. So that's one should be looked at. There are possible reasons which can be found on this page. Let's check. There are possible reasons here. These are the ones that have been listed. I'll leave the link to this page in the description box so you can read more about it. For our data, we are dealing with RNA-seq data. All right, so from the manual, it tells us this is to be expected when you are dealing with RNA-seq data. Let's check if we can find that information here. Yeah, so we have this here. So with RNA-seq data, it's observing this wobbly lines, okay, this kind of bias, especially at the start of the read. It's to be expected. It's observation that you see, or it's common in RNA-seq data. So once we see this, since we are dealing with RNA-seq data, we can proceed because this observation is to be expected in RNA-seq. If you are to deal with other data types, then you have to look into this observation. You have to look into this issue and then see what can be done about it. You can consult your sequencing facility or bioinformatician or any expert who can guide you, or just look at the manual here to learn more. All right, there's also another explanation here. So basically everything you need, once you have it, just contact your sequencing facility or whoever is in charge and they will guide you to resolve those issues. And that is the essence of doing quality control, to help us to identify issues and then resolve them. And some of these issues, it's possible that these are observations that are to be expected in the type of data. So for those ones to, you know what to do. Okay, now let's look at the sequence duplication levels also here. We'll click it to see what we have. So with the sequence duplication, this shows the degree of duplication for sequences in the analyzed Fastqc file. So that's what we see here. So here we should see 2 lines, blue and then red. All right, let's see if we can have one here. I think I saw it somewhere. Yeah, so we should see a blue and red, but in our data, for example, we see something like this. It's like they are all, they are there, but it's like they are all kind of in the same region. That's what I'll see. So here, the sequencing duplication here, the manual tells us that if we have spikes, like what we see here, if we have spikes in the blue line, okay, here we even see it in the red line as well. So if we have such a situation, it's because of a high duplication rate, so i.e. More than 50%. All right, again, it tells us, the manual tells us that this observation, when we are dealing with RNA-seq data, is to be expected. All right, so in this case, we are dealing with RNA-seq data, so it's to be expected, so we can proceed with our analysis. Again, I'll leave the link to the manual the Fastqc manual for those who want further interpretation. You can also check online for further interpretation and then relate that to the data that you have. All right. So you should take note of that. And you should also note that we are dealing with a subset of the data. And so it's also possible that that could also influence some of the results we are seeing here. All right. So ideally, if you have the entire data, then we can discuss more about it. So that's it, by the way. So there's an introductory video anyway. So it's to help you to also get started. So far everything is okay. And so we can proceed. Now, if you look at the per base sequence quality, I'll just go with that. Also, that one to the quality, we have good scores. So that's why we see them. We see the green color and we also see the check, okay, symbol. And you can see all of them in the green region, indicating high scores. All right. When we come here, it's tried to deteriorate a bit. This is also something to be expected when dealing with Illumina reads. And so what you can do is to do trimming. Okay, so that's something that you can do. In this tutorial, we are going to skip the trimming aspects, okay, and then proceed with the mapping itself. But it's something you can also do to improve the quality of the reads. All right, so for trimming, I have a couple of tutorials also. I'll leave those tutorials in the description box. There are several tools available for trimming, so you can just choose the one that you want. All right, so this is it for the quality control. You've examined the report for one file, and I am going with the assumption that we will have a similar observation in the other files. All right, so that is it. And you notice that we have several Fastqc reports, and so that is why we did the multi-QC to aggregate everything so that it becomes easy to even compare. All right, so there's the multi-QC report. You can just open it, and when you open it, you will see all the files. And you're also going to see the reports, okay, for the different files, and you can also compare them. All right, so in some cases too, aggregating them will help a lot to make your life easier. All right, so this is it for the multi-QC. So now we can proceed. We'll get back to our terminal and then proceed. So this is what we are going to do. Let's clear the screen first. So now that we have the quality control done, we can now proceed. So what we are going to do next is to perform our mapping. So we are now going to map our reads to the reference sequence. All right, so that's what we are going to do. So for the mapping, what you should note is that, anyway, let's do an ls first to check them out. So for the mapping, what you should note is that we have our data. So let's ls into Fastqc star.fastqc.gz. This gives us our data. We need our reads, which we have, and we also need a reference sequence. All right, so this RNA-seq HISAT2 require the Fastqc file for the reference sequence. So if you ls into ref directory, you will see the Fastqc file, which is there. We also have the DTF file, which is needed. So HISAT2 is a split aware aligner, and so when we supply DTF files, it is able to map to the splice junctions specified in the GTF file. So this has its uses, for the downstream analysis. Some tools, okay, for example, feature counts, which we can run before doing your gene expression analysis, requires assigning aligned reads to genes. So if we have this information captured in the BAM file, then it becomes easier to get that information. So it's usually recommended that when you are mapping your reads with splice aware the mappers, you also specify DTF file. So we need to have that as well. All right, so that's what we have for the reference sequence. So we have our Fastqc files. We also have our reference sequence. Okay, now once we have the input files, we can proceed. All right, so we have our Fastqc files. we have our reference sequence, we have the Fastqc file, we also have the GTF file. So that means we can now map our reads, okay, using HISAT2. All right, so before we do the mapping, it's important to also take care of some things. We need to do indexing, all right? We need to do indexing. We need to also make some information available. So information such as splice junctions, let's say splice sites, and then exons should be made available. All right, so we will start with that preparation first. Okay, so first of all, we will create a directory called index. So we will say make the index. All right, and now that we have this directory created, we are now going to extract some information. Okay, so we will run HISAT2. So we will start with extracting the splice site. So what we do is to say extract splice sites, and then we will specify the GTF. So we say ref/chrx.gtf. Then we will save the information to the index directory. I repeat, we have to save that information to the index directory. And so that means we have to specify that. All right. So we will say index/chrx.ss. That's for the splice site. So we will extract that information. Then we're also going to extract information for the exons. So we say extract exons.py. Then we will specify the GTF again. We will direct the output to index/chrx.exon. All right, the names have to be descriptive. So we have this also here. So we will execute this command to also extract that. Now, if you do ls into index, you will find the files there. You can even do a head index chrx, let's say exon. You find some information there. You can also do same for the splice site. You get some information there. All right, so we have that there. Now let's clear the screen. Let's ls into index and let's proceed. Now we have all the necessary files. So we are now going to build the index. So we will say HISAT2 -build -p for number of processes. We will use 8. Then we will specify the splice sites. We will say --ss. So this is index/chrx.ss. Then we need the exon, so --exon, and then we specify the exon, which is also index/chrx.exon. Then we need to specify the reference file, okay, the reference sequence and the output files. All right, we need a prefix. So the reference Fastqc file first. So that's ref/chr.fa. That's the Fastqc file. We need the Fastqc file. And then we need the output name. So the output name, we will say index/chrx_tran. I'm using the name that was in the paper that published the data. So we are using that same name. So here, what will happen is that we are going to have several files generated, and this will be the prefix, the name that will be given to these files, in addition to some other names. So all these files will have this prefix. That's the name, the base name, which is this, in addition to whatever name that was given to them. Now, you may be wondering why we did not specify the GTF. The information we need from the GTF, we have extracted them into this and this. So that's why we don't specify the GTF. for star aligner, for example, you need to specify the DTF. For HISAT2, we don't need to specify that. So we extract the information we need from the DTF separately, and then we include that information when we are going to build the index. So that is what we have. So this is HISAT2. So let's execute this command. All right, so once you execute, you will see these coming up. Those are the processing activities. So we are going to generate this file. So notice we have output files, chrx underscore tran dot star. So we will have files generated, and these files will have the prefix in addition to these other names. So we just have to wait for everything to get done for us. And take notes, we specify -p8. If you have more processes, you can increase this number to reduce the analysis runtime. All right. So if, let's say, you are using an HPC system which has more cores, then you can change this to 8, 10, 20, 30, whichever one, to reduce the runtime of this tool. So just take note of that. For those using your ordinary laptop, 8 should be fine. Okay, so you can just maintain the 8, or slightly lower, that should also be fine. So let's wait for the indexing to get completed for us. It should be done very soon. All right, so the indexing has been done. Let's clear the screen and proceed. So if you ls, we have our directory called index. And if you ls into it, you are going to find the files generated. So we have all these files. So once we have this, that means we can now proceed. So now we can continue with the HISAT2 mapping. Now, if you take a look at the Fastqc directory, let's say star fastqc.gz. All right, we have our Fastqc files. Okay, so we are going to start with just one sample first. We will do with just this one, this particular one here. Okay, so what we are going to do is to just run HISAT2 on this one, and then later I'll give you a script that allows you to run all these other files. All right, so what we are going to do is to run HISAT2. Let me just put it that way. So we have our index built for us. We have our Fastqc files. So what we are going to do is now run HISAT2 to map the race to our genome, our reference genome. So we will do with just this one, this sample here. We are dealing with paired and read, by the way. So you have read 1, read 2. So to run HISAT2 to map your reads, RNA-seq reads, we say HISAT2 space. We say -p, and we specify the number of threads. I'll use 8. All right. You can increase it if you have more computing resources. Then we will specify the index. So we say -dta. All right. So what you should note is that the -dta here is used to tell HISAT2 to report alignments that are compatible to transcript assemblies like string type. So that's what we have here. All right. Again, I'll leave the link to the HISAT2 manual in the description box. So if you are going to use string type to assemble your transcript, then that just DTA is needed to put it in the right format for you, basically. All right. Here we are only doing the mapping, so you can decide to omit that. That is fine. Okay, so we are now going to proceed. So we are now going to say -x to specify the index. All right. So we specify index with the -x, and then we will say index /chrx underscore tran. So here we don't need to specify all the files. We need just the prefix, the one that we specified when we were building the index. That is what we need here. All right, so that's what we are. So HISAT2 automatically gets the rest of the files, all the files basically, and use them. Now we need read 1, read 2. So -1 for read 1, that's forward. So we will say Fastqc /ERR 188044 CHRX1.fastqc. That is for read 1. Then read 2, that is -2. The same file, and this time the read 2.fastqc. So we have that also here. So this is it. Then we also need to specify the output file. So HISAT2 will generate our sum file. So we need -s. So it's when you get to this page, it tells you everything you need to know. All right. So you just have to make sure you check that out. All right. So sum, yeah. So basically, that's what it does. Okay. So we use -s to specify the sum file. So we will say -s, the s should be uppercase, like what we see here. That's -s. And then you specify. So we will say ERR 188044. And that's called CHRX. Or you can even choose to leave it. We can just use the sample name. That's fine. Okay. And then we say .sum. So the sum here, it should be lowercase. So let's execute the script. So once you execute it, HISAT2 will do the mapping and it will generate the output for you. So let's wait for everything to get done for us. Nice. So the mapping has been done. And so once HISAT2 does its work, it also tells you the overall alignment rate. So we have alignments, we have these percentages, or these statistics, I will say that way. And then the overall one is given to you, says 95.57%. So that means for the reads, we have 95.57% of the reads mapped to the reference sequence. So this is good news. So we can proceed. So now let's clear the screen and let's do an ls, and we're going to find a file here, which is a sum file. Now this sum file is very heavy, and so it's usually recommended to convert it to a BAM file before you can use. All right. So to convert to a BAM file, we use some tools. So we can say some tools view, we specify the file, we say -o, and we say ERR 188044.bam. We are using the same sample ID, but then we change the extension to .bam because we are changing, or we are converting the sum file to a BAM file. BAM files are relatively smaller, and so we usually do it this way to conserve space, basically. So let's execute this command, and once we do, some tools will convert the sum file to a BAM file for us. So that's how we convert a sum file to a BAM file using some tools. It's very, very straightforward. Nice. So we have the BAM file generated. Let's do an ls, and we are going to find it there. Now, some tools also require that you sort your BAM file for downstream analysis. Some tools require that. And so when you are going to do a downstream analysis, it's important to make sure you check what input you need, and you can now sort. Okay, so you can sort by name or by coordinate. There are other criteria. All right, so that is beyond the scope of this tutorial. But if there's the need to use such tools, I'll also let you know how to sort them out. For this tutorial, the focus is to show you how to map your reads using HISAT2. But if you still want to sort, you can still do the sorted one. All right. So once you have converted your sum file to a BAM file, you don't need the sum file again. So you can remove it. That's how you can remove this file. All right, so once you remove it, you have your BAM file, and whatever you want to do, you can still use your BAM file to do it. All right, so this is for one particular sample. Now, ideally, you should put your files into distinct directories. All right, so this is a BAM file. So ideally, just as we have the Fastqc file in the directory called Fastqc, we should create a directory and then put the BAM file into it. So I'll say make the mapped, and I'll move the BAM file into it. All right, so I do an ls. It's now cleaner. I do an ls into mapped. I see my BAM file. All right, so it's now very clean. So there's also a way to organize your file so that you can also do some additional processing. Okay, it makes your life very easy later on. All right, so this is it. Now you notice that we only map just one sample. Let's see if I can get the Fastqc files. Let's see. Yeah, we only map just the sample. If you have multiple samples, it will be good to have a script that does it. For this tutorial, I've prepared a simple script to do the mapping for us all. So if you want to do it, it's this one, the map.sh. Let me play the screen. Let's just do ahead. Map.sh. So this has some commands. All right, we also see them here. Let's check. It's on the GitHub repo as well, but you don't need it because it has been downloaded for you. So this is it. Okay, so I prepared this one here. Now take note that this script first creates a directory called mapped, and then it does the mapping and places the final files in those in that directory. All right, so there's also alternatives to handling multiple files. So this approach can be cumbersome for beginners. There's also a way to automate it if you have multiple files. All right, so if you have such a situation, you can also book a session with me and then we can look at that. You can also use Nextflow, by the way, to automate your scripts and all that stuff. So there's something that you can also do. All right, so there are ways to go about it when you have multiple files, when you have multiple samples. So in those cases, you can check it online. You can just send me an email. You can book a session with me and we can look at all those ones together. So what you can try is to run this entire script to get all the files, and then you can learn something. So this is for the mapping. All right, now it's RNA-seq. So if, let's say, you are interested in, let me clear the screen. If you are interested in, let's say, generating read counts, then it's important to also make sure you have appropriate tools to help you do that. So for read count, we have tools like feature counts. We have tools like ATSeq. There are other tools as well. Okay, so those ones, you need to know which inputs they require. Some require BAM files, some require the Fastqc files themselves. So you need to check all those things and then do. So if you want to learn how to generate read count for gene expression analysis, then you can also check the video right here.